as you said, I'm going to look at a particular probiotic, Bifidobacterium Breve NCFB 2258. So this was discovered here in Chagas. Our colleagues in Chagas found this. And we're going to look at this specifically in terms of mechanisms, at how it signals across an intact barrier. Okay, so as we've heard today, there's been an explosion of studies relating to the microbiome. We know that changes, or we can see that changes in microbial composition have been linked to changes in host behaviours. Some have gone so far as to say that microbes manipulate the brain behaviour and physiology to their own benefit. We have seen certain profiles are linked to a variety of different changes in host physiology, so changes in metabolism, gut function, and cognitive function. But what is eluding us is the molecular and cellular mechanisms that underlie these, this inter-kingdom communication. So we know that the gut bacteria have evolved with the host. We believe that they can communicate in health as well as in disease, and we're going to try and look at this in a healthy barrier. So within the lumen, then we have a variety of microbes. Bacteria, the most dense microbe present, particularly down in the distal colon. And our studies have been carried out in the distal colon. But we also have fungi, viruses, archaea, bile acids, and undigested foods, including some undigested lipids. Now, we've heard earlier possible mechanisms include leaky barriers, and we know that this is associated with disorders such as obesity and gastrointestinal inflammation. But as I said, there must exist a mechanism within the healthy gut where we have an intact barrier, where microbes aren't gaining access across the barrier into the internal milieu. So this must exist to facilitate cross-barrier and inter-kingdom communication. So the probiotic we're using today, it's, as I said, it was discovered here in Cork, a gram-positive anaerobic bacteria isolated from the infant gut. But what we know about it is that it produces these polyunsaturated fatty acids. So omega-3 and omega-6 are polyunsaturated fatty acids. They are essential fatty acids, so they're not synthesized within the body. They need to be taken in the diet. But they do have beneficial effects, and we've seen this in the gut, in the cardiovascular system, and in terms of brain health. In the gut particularly, we have seen anti-inflammatory effects, and we also know that omega-3 decreases GI permeability. So again, hopefully implicating a different mechanism in the signalling across the intact barrier. So PUFAs regulate gene expression. They do this by gaining access through the cell wall. We think in the distal colon this is probably likely to be a passive process, so lipids moving through the lipid membrane, because there are fewer transporters down the distal colon. Once they gain access, then they can bind to the nuclear PPAR alphas and induce transcription. Okay, so the aims of this project were to determine if luminal PUFA-secreting Bifidobacterium breve can signal across the rat colonic barrier to stimulate both local neurons, and we're going to look at the submucosal neurons because they're in closest opposition to the mucosa, and also afferent or sensory neurons, which may be part of this microbiome gut-brain signaling axis. And we want to understand some of the cellular and molecular mechanisms underlying this microbe-to-host nervous system communication. We used three different methods, but two to look locally. So we used immunofluorescence, and really key to this is the dissection. So we have applied the probiotics to the mucosa, as you would see normally, and then we remove the mucosal layer to expose the submucosal neurons. We fix at that point immediately, and then we've looked at expression of CFOS, so an early transgene, an indicator of activity within the submucosal neurons in response to the probiotics. We also carried on from there and looked in ex vivo tissue. So here we've carried out a hemidissection, so we've removed half of the mucosa, we've left half of it on. We apply the probiotics to the mucosa, and then we record the changes in intracellular calcium in the submucosal neurons. Okay, so here are some of the results that we have. So this is immunofluorescence work. We have CFOS in red, and we're looking for nuclear expression of CFOS, and we have PPAR alpha in green. So we have a number of different controls here. We have saline, so nothing has happened in this dish. Then we have applied the cryoprotectant, so trehalose, which we use to preserve the bacteria. Then we used culture broth, so broth that's had no bacteria present in it. And we didn't really see much change in terms of C-phos or in terms of changes in fluorescence of P-para-alpha with these controls. We applied the bacteria themselves, the wash bacteria, so without any of their secretions. And again, we saw little or no change in C-phos nor P-para-alpha. And then finally, we applied the supernatants, so the secretions from these probiotics, which we presume contains the polyunsaturated fatty acids. We did see a number of cells with nuclear C-phos, although it didn't quite reach significance. But we did see a significant increase in the expression of P-para-alpha in these cells. We went on then to use this hemidissection that I described already to look at calcium imaging within the submucosal neurons. So we're imaging from here and we're applying the supernatants to the mucosa. So we used broth as our control. We did get a small increase, but it returned to baseline pretty quickly. And as you can see, it's not comparable to the response that we get with the supernatants, which is a pretty robust increase in intracellular calcium, although it's quite short-lived. To determine the importance of the P-para-alpha receptors, we used P-para-alpha antagonist, and we suppressed that response significantly, although it's not completely abolished. And then to further support the importance of P-para receptors, we used a P-para agonist, and in the pooled data, we saw that there's no significant difference between the P-para agonist and the supernatants. So we went on then to look to see whether sensory afferents may be stimulated by this probiotic. So we have developed this recording technique where we keep the distal colon and all its nerves intact, and we were actually able to record from the vagal nerve up at the level of the esophagus. So first of all, we wanted to ensure that these nerves were intact, that we were getting signaling all the way up to the esophagus from the distal colon. So we stripped away the mucosa, exposing the submucosal neurons, and we depolarized these neurons. First of all, potassium chloride, and we saw an increase in vagal firing, showing that we can stimulate in the distal colon and record from the vagus nerves. This is the first time that this has been shown. And we can also do that with the cholinergic agonist carbachol. So onto our probiotic then. We have the mucosa left on in this case, and we're recording from the vagus nerve. The control, the trehalose, the cryoprotectant, does nothing, nor does the broth. But when we apply the supernatants, the secretions from B. breve, we got a robust increase in firing in the vagus nerve. We carried out the inhibition with the P-para antagonist once again, and we suppressed that response, although we didn't completely eliminate it. And the P-para agonist similarly evoked vagal firing. And again, that wasn't different. Sorry, it was slightly smaller than the supernatant. Finally, then we put on B. breve, so the washed probiotics. We haven't seen any effect locally in the submucosal neurons. But in this case, we do see an increase in firing, although it is significantly smaller than that evoked by the supernatants. So to summarize, we have shown that when you apply B. breve supernatants, the secretions from this probiotic, to the mucosa, we see an increase in nuclear C-phos and an increase in P-para-alpha expression in submucosal neurons. We can also stimulate a robust calcium response in a subgroup of these neurons, so about half of these neurons respond, and we showed that that was mediated, at least in part, by P-para-alpha. And then finally, we saw that both supernatants and the bacteria alone stimulate vagal firing. And again, this is mediated in part by P-para-alpha. So we have seen that luminal microbial products can signal across an intact epithelial barrier to stimulate both intrinsic and extrinsic neurons. Now, we don't know the mechanism here. Are these polyunsaturated fatty acids going paracellularly? Are they going transcellularly? Are they directly activating these innervating fibers, or are they relayed through the enteric neurons? But what we have shown is bacteria-derived PUFAs facilitate inter-kingdom communication, and this is a potential mechanism underlying microbiome gut-brain signaling. So just finally, to thank the people who carried out this work. This is primarily Dr. Maria Buckley's work, and Anya Lee carried it out as part of her fourth-year project. I want to thank Professor Stanton in Teagasc for providing the probiotics, and also all our funders who supported this project. Thank you.